Assalamu alaikum students. The topic for today's discussion is the lipoprotein metabolism. The lipoproteins are the spherical macromolecular complexes of the lipids and proteins. As the name indicates, these are composed of the lipids and the proteins. In this video, you will know the metabolism, formation or synthesis of the lipoproteins along with the clinicals associated to the lipoprotein metabolisms. So the major plasma proteins are the cholesterol, triacylglycerol, glycerides, phospholipids and free fatty acids. These are the major plasma lipids. Uh, the function of the lipoproteins includes the transporters. These are the efficient transporters. They provide efficient mechanism for the transport of the lipid contents toward the tissues from the blood or the from the tissues to the blood. So the lipoproteins molecules are the transporters. So the lipoproteins includes the different molecules as these are the chylomicrons, VLDL naming very low density lipoprotein, IDL intermediate density lipoproteins, and LDL low density lipoprotein, and the number fifth is the HDL, that is high density lipoproteins. These are the classification based on the density along with the protein contents, as the chylomicrons are the least dense because the protein content in them is very low. So if any one of them is high with proteins content, that will be high density. So these are the lipoproteins. And then the classification of the lipoproteins includes three types of classification as based on the density, size and lipids content, and number two is the based on the electrophoretic mobilities, that is one of the separation techniques. So ultracentrifugation is one of them. Based on the electrophoretic mobility, lipoprotein can be classified. And number third is the based on the apoproteins. Apoproteins includes the A, apoprotein B, and apoprotein C, etc. So these are the classification of the lipoproteins. Next, moving on the apoproteins. Apoproteins are the associated molecules with the lipoproteins. They have diverse functions. Apoproteins have diverse function and they are associated with the lipoproteins. They are activators for the enzymes involved in the lipoprotein metabolism. They can be act as a coenzymes. They provide recognition sites for the cell surface receptors. Apoprotein functions includes they are activators. They provide recognition sites for the cell surface receptors. And if we classify the apoproteins, the classification of the apoproteins includes apolipoproteins, that is apo C1, apo C2, etc. apo C3 is also included in it. Then moving on to the major lipoprotein classes, the number one is the chylomicrons. Chylomicrons are the exogenous genus lipoprotein that is carrying by the dietary triglycerides. Uh, Chylomicrons are the exogenous genus lipoproteins carry dietary triglycerides. The synthesis of the chylomicron includes the chylomicron assembly. It occurs in the intestinal mucosal cells where the dietary triglycerides, phospholipids, cholesterols, esterified cholesterols, fat soluble vitamins, they are assembled for the formation of the chylomicrons, as the chylomicrons is the transporter of the lipids proteins. Apolipoproteins, that is ApoB, organizes assembly B48, which is smaller and is present in the intestine. So the B48 is smaller and is present in the intestine. Now the summary of the chylomicron assembly is here diagrammatically. The lipids dietary triglycerides is absorbed in the intestine. Here it is the intestinal lumen. The dietary triglycerides is absorbed and assembled in the smooth endoplasmic reticulum. From here, smooth endoplasmic reticulum. The second number is rough endoplasmic reticulum. Here the ApoB48 is synthesized, and the ApoB48 plus the TG triglycerides, they mix up and operated to the Golgi complex. Here they are modified and due to the formation of the vesicles in the form of vesicles, the chylomicrons is excreted to the lymph, where the lymphatic vessels carry this chylomicron, and now the assembly of the chylomicron end up. Here typical clinical is associated with it, that is the beta lipoproteinemia. Beta lipoproteinemia is the condition which is due to the deficiency of the MTP, is a transporter protein, that is microsomal triglyceride transfer protein. Microsomal triglyceride transfer protein deficiency leads to the a beta lipoproteinemia. Here you can see diagrammatically that the ApoB48 is synthesized on the ribosomes present on the RER. They then transfer to the toward the ApoB particle with the mixing of the lipids, and this mix up is carried out by the MTP, microsomal triglyceride transfer protein. If the deficiency of the MTP occurs, if defective, no proper transport occurs, and no assembly of chylomicrons occur, and this condition present a beta lipoproteinemia. Now moving towards the transport of the chylomicrons, that is from the dietary lipids, it is absorbed from into the intestinal cell. Then the intestinal cells chylomicron with the mixing of the apoprotein C, A and E along with the B48 apoprotein B48, they mix up, and then the chylomicrons is not directly transferred to the liver but toward the blood capillaries, mostly the capillaries of the skeleton and cardiac muscles. Here the lipoprotein lipase is activated. Lipoprotein lipase is activated by the chylomicron. Lipoprotein lipase activated, and then the chylomicron remnants produced, as the the glycerides and the other fatty acids are removed out during lipoprotein lipase action. So the chylomicron remnants produces these remnants, then transfer to the liver by the remnant receptors present on the liver. So this is the five steps by the transfer of the chylomicron. And here the important chylomicron point is that nascent chylomicron. Nascent chylomicron is the particle released by the intestinal mucosal cell, which is called nascent chylomicron. Because it is present, it is incomplete, so it is called nascent chylomicron. Here the some functions of the lipoprotein lipase is that It is the extracellular enzyme that it degrades the triacylglycerides contained in the chylomicron. It is anchored to the capillary walls, mostly of the cardiac and skeletal muscles. It is activated by the ApoC2 on circulating chylomicrons, present on the circulating chylomicrons, and it hydrolyzes the tag toward the into the fatty acids and glycerols. Fatty acids and glycerols then moves to the other metabolic pathways. Here, the clinical condition associated with it is the type 1 hyperlipoproteinemia. Type 1 hyperlipoproteinemia, it is the lipoprotein lipase deficiency. The ApoC2 may be defective in this condition, and here the chylomicrons triglycerides accumulates in the plasma. The chylomicrons levels increases highly elevated in the plasma. Then the treatment includes the reduction of the dietary fat of the type 1 hyperlipoproteinemia. So this is the end of the lipoprotein metabolism part 1.